later part of presentation. So now coming to part 2, and now I would introduce you to this exciting tool, CRISPR-Cas9, and how these tools is being used worldwide in different sectors to develop precise variation and to improve crop in a very rapid pace, which would not have been possible earlier with earlier technologies that I discussed. So if I want to just represent the CRISPR-Cas9 to you in a very layman language. So it has two systems. One is customizable RNA, and another is a protein that's called Cas9. So they're like this customizable RNA. You can develop a design RNA. These RNA actually, the RNA that would be homologous to the sequence that you want to change, to the DNA that you want to change. So this is the address, genomic address, the address where you want a change in your genome. And this Cas9 is like a, if I want to give you an analogy, this Cas9 is like a postman. So it has two scissors. One is called RubC and another is HNH. These two are nuclease domain of this Cas9. So this postman has two scissors, just keep in mind. And another, this customizable RNA is the address that you are giving to the postman. So postman takes that address with it, and it goes to the genome that you want to modify. And then it go and it searches for the homology of the RNA sequence with the genome sequence. So when the RNA sequence exactly match, a 20 base pair RNA sequence exactly match with the genome sequence, then the Cas9 or the postman gets a signal that this is the address where I need to make a cut, because it has two scissors, I told you. Now, after it finds this match, it binds perfectly with the genomic DNA, and then it makes a double strand break in the targeted gene or targeted DNA with its two scissors, okay? So now, whenever you make a cut in your targeted gene, then the gene becomes non-functional in most of the cases. Now, what happens? Once you break or once you create a double strand break in the genome, what happens to the genome? What happens to the DNA? You know, DNA is very, very crucial part of any cell, any cellular organism. So whenever there is a break or damage in DNA, the cell takes it very seriously and it immediately starts to repair it. So how to repair the two broken, and they want to rejoin perfectly to make the one which was originally there. So there are two repair mechanisms in cellular system. One is NHEJ, another is HDR. But the NHEJ is a predominant method of repair in any eukaryotic, higher eukaryotes. So this NHEJ is also a little bit error prone. So when they repair the two broken end, they randomly insert or delete new nucleotide in that cut point. So you see this red color is a random insertion or deletion of one or two letters of the DNA, the nucleotide of the DNA. And because of this new insertion or deletion of one or two letters from the gene's sequence is gone. So the open reading frame of that gene is altered. So this gene would not produce the same protein as it should produce in the original condition. Now this gene is non-functional. So this gene is knocked out. So you disabled the gene because of creating a targeted break and the cellular repair system, okay? Another system is homology directed repair, but that is very, very less efficient, and that happens in cellular system rarely, normally during the time of in dividing cell or during meiotic recombination. So what they do, they repair the same break perfectly by seeing the sister chromatid as a donor template. So in any diploid organism, there are two copies of one chromosome. So suppose one chromosome, one gene is broken, one copy of that gene is broken. So another copy of that gene is intact. So the homology-directed repair, what they do, they see the intact piece as the template, and then they copy the information into the broken piece, so making it a perfect repair. But that is very, very low in efficiency. So if you want to use homology-directed repair in order to repair your gene of interest, then, and you want to incorporate some kind of special changes in base pairing, in base sequences, or you want to insert or delete something exactly, not the random one. So you have to give them a donor template with a homologous end flanked to your intended change. So if you see here, the blue is your change that you want to incorporate. And you, in that donor template, you make two homologous arms. And then the cell will treat this donor template if it is present in adequate amount. A cell will treat it as a template. So they will copy this information to the broken end, okay? And then your intended change is incorporated into the genome. So that's the homology-directed repair. Now, you would be clear from these video narration.
Been adapted into a powerful tool for genomic research. There are two components to the system, a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay.
Been adapted.
Okay, so now this CRISPR-Cas9 also allows us to deactivate or disable multiple targets or multiple genes in one experiment. So you don't have to go for one by one. So you can design one single experiment and you can target as much as 8 to 10 targets in a single experiment. So how it could be done? So you see, I told you that there is a two-component system. One is Cas9, another is the address RNA, that is called guide RNA. So if you design multiple guide RNAs and give it to Cas9, so this Cas9 takes one guide RNA, Cas9 takes another guide RNA, and this Cas9 takes another guide RNA. And this complex of Cas9 and guide RNA, they go to their designated targets. Like this one goes to gene 1, and this one goes to gene 2, and this one goes to gene 3, and likewise. So you can do editing, or you can disable multiple genes in one single experiment if you have multiple targets to disable. Okay, so multiplexing is easy with this system. So now I want to just briefly tell you about these three systems. One is, I explained to you that already double-strand break is possible with Cas9 and guide RNA. It has two Scissor, and then it has, you can engineer that Cas9 to make it a single strand break. So it is called Cas9. So the wild type Cas9 has two Scissor, okay? It has two nucleus domain. So now if you engineer that Cas9, you can make it the Cas9 with a single Scissor. So it will make a single cut in one of the DNA strand. So it is called Cas9. You can make it as a dead Cas9, but disabling both the Scissor. So it is called dead Cas9. So Cas9 has two properties. One is binding to the exact position where the RNA actually directs, and then it makes a cut. So it has two properties. One is binding to the target sequence and then cutting the target sequence. So the dead Cas9 can bind to the target sequence, but cannot make a cut to the target. So I'm not going details into all the CRISPR-Cas tools that is available for all kind of biotechnological applications, but yeah, I dealt with a webinar a few days ago, so you could go through the YouTube and you could listen to me for knowing all different kind of tools, but I'm not going into details today. So in part 3, now this is the part where I want to justify my title, that how it is rapidly accelerating the crop improvement. So I would give you some examples of how it is impacting crop improvement all over the world. So, you know, this took 9,000 years. Coming from teosinte to modern day corn, it took 9,000 years to develop by different kind of selection, artificial selection, to develop modern day maize, to feed the growing population. Now, this CRISPR-Cas tools is amazingly accelerating the domestication. So the wild crops could be domesticated very, very fast manner by using CRISPR-Cas9. So if you say this is an example, classical example of ground cherry, and this ground cherry, if you say this is the wild type ground cherry, which has a bushy nature and which takes a lot of space to grow. And one of the domestication characteristics is to develop that plant to a very compact, organized plant. So by knocking out those genes that are responsible for domestication, because we already know the genes that are responsible for domestication by your basic studies, then you target those genes to edit, and you make in 2 or 3 generations, even in 1 generation, a compact ground cherry plant that looks like a domesticated one. So this is another case where they knocked out another gene for fruit number. Those ground cherry actually has a very less number of fruits, but you see here, the fruits per 5 nodes has been significantly increased in the edited one. So you got to compact the stature of the plant, and then you increase the fruit numbers. And then if you see here, the flow rate number has been increased 5 flow rates to 7 flow rates, and then locule has also been significantly increased from 2 to 3 or 4. So those are all happening in 1 or 2 generations by the application of CRISPR-Cas9. So you don't have to wait for 9,000 years to domesticate them by artificially, phenotypically selection, generation after generation. But in this CRISPR-Cas9 approach, so that was the classical example for a rapid domestication of ground cherry into an edible one with all the domestication characters. So now, if you see, this is a conventional tomato that you look for when you go to a grocery shop. But you know, always my father, my mother, and even my grandfather, they used to complain that those tomatoes are not that tasty that we used to have in our childhood. They always complaining, you know. Why? Because scientists and farmers, they always go for selecting the fruit size, fruit number, and other characters that would give them more yield and more benefit, and they have not probably focused on the taste of that one. So it is not as tasty as it should be, or as it was in the earlier cases. So this is another extraordinary example of domestication. This is a Solanum pimpinellifolium. This is a wild tomato. It is very tasty, but the size is very small, and it grows in very small number. So if you see, there are 2 back-to-back papers in Nature Biotech recently that they have independently domesticated this too. They have independently domesticated this Solanum pimpinellifolium into domesticated crops. So if you see, this is the wild type, one, Solanum pimpinifolium, and it is growing like bushy and taking a lot of space. But what they have, when they have mutated ASP gene, that is a gene responsible for domestication, then they got a compact plant, you know. And this is a nice picture from that same paper, that this is the wild type one, that inflorescence. You can see the inflorescence here. It has the number of flowers in inflorescence is very less, whether the one which is edited with CRISPR-Cas9 has significantly higher number of flowers in a single inflorescence. So if you all see here, I told you that Solanum pimpinifolium is very tasty, but it has very small fruit size. So if you see, the fruit size has already been increased with this CRISPR-Cas9 application in that species. So the fruit locule number and fruit weight has already been also been increased by this rapid domestication program. So now this is another extraordinary example of using CRISPR-Cas9 for humankind, and that has not been possible earlier. You know, if I talk about hybrid seeds, and many ask me, and many people ask me, okay, Kutub, why farmers need to buy every time the hybrid seeds from the company? Okay, and it is, they cannot save their seeds for the next generation, because, you know, hybrid is a technology when the two parent hybridized and F1 hybrid develops. This F1 is normally, this is called heterosis or hybrid vigor. This F1 or hybrid plant is more vigorous in almost every sense than their parent. And this first generation only gives the yield advantage over the parent. But if you save the hybrid seed and for the next generation you grow them, then the character becomes segregated, and you won't get an homogeneous yield or homogeneous higher yield of that hybrid. So every time you have to, if you want to grow hybrid seed, every time you have to go for purchasing seed from the company, especially in case of rice. So there is two papers, one is in Nature and there is Nature Biotech. What they have done, they tried to develop a hybrid seeds that could be clonally produced. So theoretically, that seeds, if it comes to market, it could be saved by the farmers to generate year after year, and they don't have to go to buy every year, okay? So what they do, if you see, this is a hybrid seed, this is diploid hybrid, where two components, red and blue, coming from two different parents. So that's why they are hybrid and they are more vigorous than their parent. Now, in normal cases, this hybrid, when they develop two gametes, one is pollen and another is ovule, and they go for meiosis, that is called reduction division. So they develop haploid gametes N and N, but what they have done, they mutated 3 genes and they induced a phenotype that is called mitosis instead of meiosis. Mitosis instead of meiosis, that is called MIME. And this MIME phenotype, what they do, so this hybrid would not go for meiosis to develop their gamete, rather it would not, it would go through mitosis to develop their gamete. So gamete would be diploid, not normally haploid one. So the diploid gamete is produced now, and we know that rice is self-pollinated crops. So the two gamete would be fused by self-pollination, and then it would develop a tetraploid plant in normal cases. But in another intervention, in the same paper, what they have done, they mutated another gene that is called not like dad. This gene is called not like dad, or matrilineal gene. So these, when they knocked out this gene, then the male gamete, the genome of the male gamete is eliminated. So only the zygote would be formed from only one of the gamete, that is from the female gamete. So you see, the same genetic makeup of a hybrid is retained in the zygote. And if you develop a plant from that zygote, then you develop a clonal hybrid embryo and a clonal hybrid plant. So it is exactly similar as the hybrid one. So if you fine-tune this technology, it would greatly impact the poor farmers in the developing countries and obviously in the whole world, because it is going to revolutionize agriculture if it comes to market. So this is how we could change the entire agricultural paradigm